Welcome to ISL-enabled virtual lab of CSIR.
It is said that the nature has invented reproduction as a means for life to move forward. As a life force that passes right through us and makes us link in the evolution of life. Rarely seen by the naked eye, this connection between us and the plant world is truly a wonderful creation. This natural connection is getting more and more stronger by understanding the magical characteristics of plants by means of wondrous innovations in plant biotechnology. It has unfolded many mysteries of plants, giving option to utilize maximum benefits of the plants. Among such magical features of plants is its capability to reproduce the same plant from any part of the plant. This ability of a plant cell is scientifically called as totipotency, and the technique used is called as plant tissue culture. The technique of plant tissue culture has been very advantageous in many ways. Conservation of endangered plants, rapid production of mature plants, production of multiples of plants in absence of seed or pollinators, production of clone copies of plants for desirable traits, particularly for flowers and fruits. Production of disease-free plants in sterile conditions and mass propagation of plants for commercial use are major advantage of plant tissue culture. With India's increasing demand for agricultural, forestry, plantation, and horticulture crops, the need of high quality, high yielding, and disease-free plantlets has also increased. Plant tissue culture has emerged as an important biotechnological and commercially possible mode to multiply elite varieties of plants rapidly in specialized laboratories irrespective of the season round the year. Farmers in India have upscaled their income due to the commercial use of plant tissue culture technique. The CSIR, National Botanical Research Institute at Lucknow, has a marvelous and rich history in the scientific developments of plant tissue culture. Since its inception, the scientists have researched plant tissue culture on various plant species and have successfully transferred the researches for India's agricultural reforms. Due to its amazing credibility, the groundbreaking success of plant tissue culture technique has been observed in banana cultivation in Uttar Pradesh. Today, plant tissue culture has emerged as an important biotechnological, commercially viable mode to grow variety of disease-free banana rapidly in specialized laboratories irrespective of the season round the year. Banana production by tissue culture plants are uniform and are large in quantity due to less cultivation time than traditional banana cultivation. This motivates farmers to adopt banana cultivation by plant tissue culture technique to remarkably increase their income. Tissue culture has now been a possibility of successful agricultural practice in India and has taken its place in India's growth, especially in farming. This success makes us curious to know the science behind the tissue culture technique. The first step in the tissue culture technique is a proper selection of a mother plant. Then the cutting out of the explant part from the mother plant is done very carefully. Proper washing of the explant is done in running water to remove mud and other impurities. It is then shifted to lab for the removal of contaminants by prescribed special solutions. Afterwards, it is being shifted in laminar air flow for further treatments. The next step is the inoculation of explant, where the plant is being transferred in customized nutrient medium. After this, the explant is shifted for culture to specialized culture room, where in vitro growth will take place in artificial conditions made as similar as natural conditions. Further, the in vitro shoots are separated again in the laminar airflow station, and these fully grown shoots are transferred to the rooting medium for root induction. The innovations at National Botanical Research Institute is now rapidly bringing phenomenal change in the lives of not only of banana farmers and has identified itself as the forerunner of modern agriculture and high-tech farming. Like such innovations, the future in biotechnology and plant tissue culture is highly advantageous for the aspiring young science students of our country.